So signaling cascade, you should know that it is the uh, sequence of events uh, in cell which is started from a receptor and leads up to the effector, because in our last lecture we have talked about this, that how this signaling cascade will start. It will
Yes.
start from the receptor,
Yes, sir.
right?
जी sir.
And this receptor will definitely get any specific stimulus,
जी.
right? And
Yes.
that stimulus will be received by this receptor, and then receptor to you have to go through some proteins. Those proteins will be on the pathway, on this path. For instance, there is a protein 1, then you have a protein 2, then you have a protein 3, and then finally uh, uh, this movement of signals from one protein to other and then other, at the end you would have something like effectors,
Yes, sir.
uh, effector, and this effector will be responsible for eliciting or for exhibiting the response,
Yes, sir.
right? And now signaling cascade is actually the sequence or the chain of events. We can easily define this by this example: chain of events,
Yes, sir.
a chain of reactions by which stimulus leads to the response, and it is not possible to bypass these steps. Any signal which is coming from outside or inside, and it is perceived by this receptor,
Yes, sir.
and then it has to go through all these steps, 1, 2, and 3. It cannot, you can say, mm, uh, jump over from these steps. It has to follow it if it is going to show any specific response. So this is what we should know about the cell signaling. All right,
Yes, sir.
are you getting this point? So
Yes, sir.
now we will discuss an example. From there you will easily understand this concept, uh, and we can start it by showing an example. uh, Maybe it is present in your book as well, like this.
Yes, sir.
Okay, for example, you have a protein, a channel protein, like here, this way, and you have a signal like this from outside. It is coming from outside. There is a, we call it protein receptor.
Yes.
Protein
Yes, sir.
receptor, and there you have a signal, and with this all you have some internal protein. Like I can say, here you have another protein like this. It is called G protein. It is called
G.
G proteins. So yes, it is a G protein. Actually, uh, the changing in the shape of the receptor, it will allow that structure, like this structure, to bind with this. Like, it is very easy to understand. For instance, you have this receptor outside. If any signal will come and it will bind here, so first the protein was like this, but once it will attach with this body, with this receptor, because this is receptor, and this receptor will change some its shape, or it will change its conformation,
Yes.
and it will combine with this, will bind with this protein that is G protein, right? But
Yes.
it is only at the time when this signal will come to bind here. Otherwise, both these structures will remain separated like here. Here
Yes.
you will see this receptor is not attached to this G protein, but it will only attach at that time when this signal will come to bind here. Are you getting this point?
Yes,
So
sir.
this is one thing that you should keep in mind. Then in a similar way, I'm here to tell you about uh, the pathway, the signaling cascade. We will try to understand by this example that how signaling cascade is actually working in the cell of an organism.
Yes, sir.
So there you would have another protein, another channel protein, or another, you can say, a messenger. It is called second messenger
Yes, sir.
because it will receive the message from which is coming from this combination. So once when the signal will come from this to this G protein, from there it will come here,
Yes, sir.
right? At this point, it is also this second messenger is actually an enzyme,
Yes, sir.
and that enzyme name is adenyl
Cyclist.
adenylate cyclase,
Yes, sir.
right? So here you have this structure, complete structure like here, like this. It is also called AMP because it is adenylate cyclase. So it makes second messenger. So it is AMP, right?
Yes,
So
sir.
here is a cell surface that we have observed here. Now, from this cell surface, when it will come to bind with this enzyme, what it will do? It will, you can say, disintegrate or to spread or to give a second messenger, like you will get here second messenger AMP. Like here you have number of activated enzymes. So all these are, what are they? They all are active enzymes,
Yes, sir.
the activated enzymes. Now these adenylate cyclase, first these were inactivated, but once this signal is coming in and it will bind with G protein, and from there it is coming toward here. So when it will come here, it will activate these enzymes like it, right?
Yes, sir.
So these activated enzymes will do what? These activated enzymes will activate final enzyme
Okay, sir.
because it is a cascade. It is the chain of events.
Yes, sir.
So signal come here. When signal is when signal oh wait, you have this. Oh, oh. So beta, this signal attached with this, it has activated G protein. G protein activated this enzyme. This enzyme activated the second enzyme, and that is the final enzyme. Or it may be 2 to 3 steps which are involved. Are you getting this point?
Yes, sir.
Right, beta?
Yes,
So
sir.
at of this, there would be a response. There would be a response,
Yes,
and
sir.
that response would be metabolic change.
Okay, sir.
Metabolic change.
Yes, sir.
So what we have learned here, this chain of events is a signaling cascade.
Yes, sir.
Let me respond this one. जी अस्सलाम वालेकुम. Hello. Sir, thank you so much. God bless you. Thank you so much. ठीक है, मैं जरा अभी मसरूफ हूँ, बच्चे की क्लास ले रहा हूँ. Thank you very much. Okay. Okay. Uh, so, uh, this is a signaling cascade,
Yes, sir.
right? Signaling cascade. What we want to understand here that there are chain of events uh, in which one enzyme or one protein activate the other protein activate another, and this is how the final response will
Yes,
take
sir.
place, right? So
Yes.
that's why we call it is the signaling cascade,
Yes,
right?
sir.
So the all types of cell signals, like we can talk about another type of another example here. Here you will understand that this is the pathway that you have like there you will understand that how uh, uh, response takes place in the cytoplasm. There you have number of things, but there would be another case that we can discuss with. Like you have a cell,
Yes, sir.
like you have this cell, right? And there you would have something inside all other structures like,
Yes, sir.
as you know, we can discuss that these endoplasmic reticulum and some other structures which are present. So in this case, for instance, we have again we have a receptor here,
Yes.
right? And this receptor would be hormone. For example, this hormone is insulin,
Yes, sir.
right? Once this is an insulin, when it will come to bind here, right? What would be the result? This will come, and here you would have a glucose, right? And this glucose
Yes, sir.
will be converted into what? जी बेटा,
सर जी,
into glycogen.
इंसुलिन आएगा तो
Glycogen,
ग्लूकोजन नहीं कन्वर्ट होगा सर जी?
नहीं, वो आप वो बेटा
वो
ग्लूकागॉन है.
वो ग्लूकोगॉन
Glucagon is a
सही है सर.
hormone
सही है,
which
समझ गया
actually
सर.
converts the glycogen into back into glucose.
यस
Here
सर.
you have glucose. So when this glucose will increase in our blood, so our blood will recognize that the blood pressure will getting increased. So
यस
in order to maintain that blood pressure, this insulin will come, and the extra amount of glucose will be converted into the glycogen,
यस सर.
right? This all is going on into the liver cells, you know.
यस
So
सर.
there you would have the, so this glucose is absorbed inside. For example, this glucose is coming from outside to the bloodstream, and this is a glucose, and that is absorbed by this cell. This cell has absorbed, because this is a cell, and it has absorbed this glucose which is present here. So when it will increase here, so insulin will come and it will convert into the glycogen. This is one. So now this is one case that you have studied.
यस सर.
There would be some hydrophobic signals. Hydrophobic signals. Intracellular, its 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 target is, for example, you have here a signal, right?
यस सर.
In previous two cases, we have studied there is a receptor which is present on the cell membrane, that is present on cell membrane. But in this case, when you would have a hydrophobic cell,
यस सर.
hydrophobic signal, sorry, hydrophobic signal. This hydrophobic signal will do what? It will come here and it will enter into the, there is no receptor here. It will get into the cell. Its receptor would be intracellular target. or its target would be intracellular. Now
Yes.
the question is, what type of signals which have intracellular targets? They don't have any target or any receptor on the cell membrane. So, for example, it's like, I can give you an example, like steroid molecules. Yes, steroid hormones.
Yes, sir.
Steroid hormones always come into the cell, and they directly enter into the lipid bilayer. For example, you have an estrogen, you have testosterone, and many other hormones which are steroid hormones. They don't have any target on the membrane. They always get into, and there they would have some target on which they will act. Right?
Yes, sir.
So this is another case, and this is for steroid hormones or hydrophobic signals. Remember, all these were hydrophilic signals that you have studied. Like they have their receptors on the cell membrane, and the cell membrane will attach. This signal is not coming in. This signal is just attached outside,
Yes, sir.
right?
Yes.
Once it is attached outside, so it will just elicit or just you can say boost or just uh, initiate the signal, and that signal will come in and it will go further. But
Yes.
this hormone is not coming in. But in this case, when you have steroid hormones, they will directly enter into the cell, and from there they will perform its function. You
Oh,
would have another case. Another thing would be like cell to cell adhesion. How one cell attached with another cell. Like these are two cells,
yes, sir.
right? So it is cell to cell adhesion. Cell to cell adhesion.
Yes.
Direct cells to cell signaling by cells in contact with each other. For example, lymphocytes detecting foreign antigens on other cells.
Yes, sir.
Lymphocytes are, defense system or immune cells.
Yes, sir.
These immune cells, they always do what? They sometimes recognize the foreign particles or outside antigens on the surface of another cell, and there it will come to bind with it. And for example, this is a lymphocyte, and it's going to attach with just to engulf it, to break it, to destroy this cell.
Yes, sir.
Because if this cell is not being destroyed, so it will create a problem in the body. So once the immune cell or lymphocytes, when they will recognize any cell that has foreign particle outside,
Yes, sir.
these lymphocytes will come to engulf it, to
Yes.
break it, to
Yes, sir.
destroy it.
Yes, sir.
So this is cell to cell adhesion sort of example that you can easily use, right? So from here, what we have learned, that there are different types of signals, and those signals have different targets. Some targets are outside of the cell or the cell membrane. Some targets are inside the cell, and specific signals directly get into the cell, and there it performs their function. Are you getting this point? Or Raise,
Yes.
I think you're getting this point.
Yes, sir.
So function of the cell surface membrane. What is the function of cell surface membrane?
Cell surface
Very
membrane.
yes, uh, definitely very simple. One is it separates the cell organelles from outside environment.
Yes, sir.
Is it?
Yes, sir.
Another function we have studied right now:
Cell signaling.
communication. Communication with other cells by cell signaling.
Yes, sir.
Why this cell signaling is? This is the language of the cells. The way I talk to you and you communicate with me, we are communicating by words. But how these cells communicate with one another? These cells communicate with the help of these signals. They have the receptors, and these receptors are their ears,
Yes, sir.
and they have other like cascade of signaling a signaling cascade. So that is actually their brain to process that information and to to lead that information to the target. So
Yes, sir.
the method of communication is different in case of cells. Another function is partially permeable membrane, allows some substances to get in and restrict some substances not to get in.
Yes, sir.
It means control the transport of materials into and out of the cells.
Yes, sir.
Are you getting?
Yes, sir.
And it is also the site of various metabolic reactions,
Yes, sir.
right?
Yes, sir.
So this was all about the cell signaling. These examples you should keep in mind. That one example you have studied about
Glucose.
the uh, you have studied about there are many pathways. The pathway involves hormone glucagon and adrenaline to release glucose in blood, depending that we have talked about. And another example you have studied that the
Insulin.
G protein. G protein activate the enzyme adenyl cyclase, which in turn make AMP second messenger, and
Yes.
that trigger the sequence of events called sex signaling cascade. G protein, in case of G protein, we have studied that signal come and it binds with the receptor. Receptor will attach with G protein. G protein is going to activate the enzyme adenylate cyclase, adenylate, what we call it here, adenylate cyclase. So this adenylate cyclase produce second messenger in the form of this. And these second messenger are going to activate other enzymes. And those other enzymes will go to activate other. And at the end, you will get a response. Right?
Yes, sir.
G protein, you have an example of G protein. Then
Yes.
you have an example of glucagon. Then you have an example of steroid hormones
Yes,
like
sir.
estrogen, testosterone,
Yes, sir.
and you should know the differences.
Okay.
This is how they are performing their function, and this is all about, right बेटा? So इधर ही कंक्लूड करते हैं. नेक्स्ट टाइम हम जो है वो आगे ही हमारे पास शॉर्ट सा पॉइंट है उसको हम डिस्कस करेंगे एंड देन दिस चैप्टर विल बी, आई विश मेरे पास थोड़ा सा टाइम जब मैनेज होगा बेटा, देन डेफिनेटली वी विल डिस्कस अबाउट डिफरेंट पेपर्स, पास्ट पेपर्स लेके जो मेरे पास चीज़ें पड़ी हैं, असाइनमेंट जो भी हमारे वर्कशीट्स हैं, वी विल ट्राई टू डिस्कस वर्कशीट्स एज
सही है
वेल.
सर.
एंड वो हम फिर जो टॉपिक लेके उसमें से भी हम उसको साथ साथ सॉल्व कर सकते हैं.
Yes,
सो
sir.
कम टू दिस प्रैक्टिस एज वेल
ठीक है सर.
बिकॉज आपके पास टाइम कम है. ठीक है.
Yes, sir.
That's all.
Okay, sir. Allah Hafiz.